All right. Awesome. So here we are again with the Matt and Pat show. We're joined by Zara and Sean. Just having another little conversation about, I don't know, what do you say, Matt? 3D bioprinting. We're talking about a little bit of Organova. We had a nice quarterly result, I don't know, a few weeks ago. And we're going to
Yep.
touch on that. And then we're going to just kind of ping pong it around and see what's new. What do you
Yeah,
think,
that's
guys?
exactly right. Yeah, I think that we should kind of talk about a couple different things. Zara's here and knows a little bit about stem cell use in tissue engineering, and then we'll slide into Organovo and bioprinting and what we know about that, and a little bit about the earnings call, because all of these technologies kind of come together to for the future of regenerative medicine, no doubt. So I'd love to hear, Zara, about your studies and what you're doing.
As you already know, I am doing my master's study in tissue engineering, and my main focus, even though I have hands in other researches too, but my main focus is to engineer full thickness human urethra and fully from stem cells and autologous stem cells, which is come from the patient. Means no other sources, no allogenic sources, no xenograft sources is involved in this project.
Mm.
So you,
So this
you're
is mainly, yeah, the area of my job.
So you're taking the fat cells of an individual and
Yes,
teasing out the cells. These
we take
are
the yes, we take the fat from patient either in the form of solid fat or in the form of lipoaspirate. Then we try to isolate the stem cells from fat. But for my project, I try to make the scaffold itself from the cells too, which is known
Wow.
as self-assembly scaffold. I don't know if you know about that or not. Then I try to differentiate those isolated stem cells to two cell types, which is needed for my that tissue to make that tissue. Then cells are there, scaffold is there, so we try to make them all together and make a intact tissue to be used for whatever surgeons for doing any reconstructive surgery that they want to do.
And then for the urethra, you're actually doing fantastic research, because I imagine this tissue will not be rejected by the patient. And
Yes, yes.
ultimately, you're
The
studying.
hypothesis is that, yes, the hypothesis is based on that. Since this is autologous, no chance of rejection. It
And
comes from the patient to the patient.
Yes, so the same genetic material from the patient, drawn from their fat
Exactly.
cells, creating the urethra tissue. This is
Yes,
just
yes,
one specific tissue, but this allows you to consider that if you use these stem cells that are drawn from the patient with the same genetic material, all kinds of
yes.
different tissues could be made all the way throughout the body, because these stem cells can be told to be any type of cell in your body. Is that correct?
Yes, right. But not that easy. I
Well, I
mean,
didn't say it was easy. That's up to
no,
you
no.
scientists.
No. In theory, they say you can convert stem cells to any. Yes, in theory, yes. But when you come to practice, it doesn't work that way. You convert them to whatever cells you want. Let's say you convert the stem cells to urethra, then you seed it on the scaffold. But after 2 weeks, they lose their phenotype, and this is
Mm,
the challenge that we are facing.
wow, that's tough.
I mean, this is not that easy. This is what I mean.
Yeah.
Yeah.
You convert, but after 2 weeks, they lose the phenotype.
Wow. Huh.
But
Yes,
That's
what an exciting science though, right? I mean, you go to work
yeah.
every day and just be like, what is going like are there new things that are popping up every day? I mean, what's
exactly.
it like at work? Like what's a typical day at work for you? You know, just
To be
something we
honest,
just invented
I
the
just
why we just broke the, you know?
know
What's it like?
this is like, okay, if I want to see say the theme of the research work is just to repeat. This is my typical day at work. Just repeat whatever I did yesterday and fail.
Oh, you
This
got to
is what
keep
we,
And then you
you
just
got to
keep
yeah,
keep
just
pushing.
uh,
ad,
keep
adverse
repeating.
adversity seeds greatness,
Mhm.
right?
That's great.
So,
Greatness. Yeah. Yeah.
I mean, you can't steal second with your foot on first.
Quick question for you. I didn't. I was a little bit late, so I didn't get your name. I think from my peripheral, I heard like Sarah or
Zara.
Yes,
Zara.
yeah.
Okay.
With a
Zara.
Zahra,
Z.
Zahra.
Okay.
Zara.
So, question about the scaffolding. You mentioned that the scaffolding itself is strictly stem cell based. Is there no
Yes.
ink or gel or anything that's initially?
No, no, no. This idea is based on, this is invented in 1970 some 1990 something by a group in University of Lava, if I'm not mistaken. So they tried to induce stem cells, not any type of stem cell, adipose-derived stem cells, not any other kind of stem cells, or dermal fibroblasts, or a smooth muscle. So they tried to culture these 3 cell types under the influence of ascorbic acid. And they find after like 4 weeks of culturing cells, they can make their own extracellular matrix, which is known as self-assembly. Means they know the architecture, the cells know the architecture, make it, and they sit on that. It is like a scaffold. You can use it as a sheet, then you sit your cells on top. This scaffold is produced and secreted by the cells and is mainly made from collagen type 1, 3, fibronectin, elastin, and many other components which is not known. So this is what, I don't know if you said your company is working for bioprinting, right? If I'm not mistaken.
Yes.
So this is my suggestion. Since this is no work done for urethral tissue engineering, so but if you want to try it for the first time, and as long as I know that you are going to make it in vitro first, then implant it inside body. So my suggestion is that you make that bio ink as much as possible close to the native urethra. But another issue here is the exact component is urethra is not known. So just go with whatever is known. So make it as much as close to it, and try to do some like preconditioning, like make some angiogenic, induce these cells to do angiogenesis and make like blood vessels before you can implant it into patient, since you are going to make it in vitro.
Yes, real quickly. Actually, we don't work for the company. We're investors in the company. But
but at least this is idea.
I like the idea. I think it's a nice suggestion.
No, it's a great suggestion. She just may not know, because this is really recent. They've printed liver tissue. So it's self-assembly by printing. And the liver tissue has been contracted out, as well as the kidney tissue that they printed has been contracted out to pharmaceutical companies for the last couple years for toxicity testing. Not only that, just in the last year, they printed liver tissue onto and grafted it onto mice. This was presented at Termis, TERMIS, which is a conference. The results of which the mice lived, it effused, and the graft took and actually showed expressions of like it was working. Markers were coming out, that human markers were coming through and passed to show that it was living. And the mice lived past 28 days. Now they're with mice that are living past 90 days with the bioprinted liver tissue. And the thought is that of moving into pediatric indication with FDA, they'll probably submit next year for pediatric metabolic disorders of the liver. So so it's that actually what you're talking about is finding the specific patient or the skin type, or that it's actually liver tissue. They found it. It is in the clinic. It's being studied. So it's unique.
Impressive, because this is usually difficult, since liver is a thick tissue, and if you can make it outside body and allow the all nutritions and the blood vessels come before you implant it inside body, this is a really big job. Means not every researcher can do it.
Yeah, the vascularization of it has always been the toughest part, and I think,
Pat, can you pull up Organovo? Maybe put mouse and Organovo, mouse, and Termis, TERMIS, and see and pull it up. Maybe we can just read real quickly, because the mice were with a diseased liver, I believe, and then the patch helped.
Mice, Termis, Organovo,
Yeah, I think Pat can bring it up on the screen.
Oh, okay.
So we could all see it at the same time, but.
Can you see it?
At the moment.
I can see it, but just barely.
Okay.
Can you read it, Pat?
I can. Here we go. Organovo presents first preclinical data on 3D bioprinted human liver tissues at Termis Americas meeting. Results demonstrate clinical promise for bioprinted tissues. San Diego, December 13, 2016. Organovo Holdings Incorporated, a three-dimensional biology company focused on delivering scientific and medical breakthroughs using its 3D bioprinted technology, is presenting the first data showing survival and sustained functionality of its 3D bioprinted human liver tissue when implanted into animal models. This data is being presented at the Termis Americas meeting in San Diego.
Okay, hold up. Now just skip the next one. Let's go. I want to go down to the third paragraph. It implanted the 3D bioprinted human. Oh, you went too far, but that's all right.
That's the second part. Okay.
Yeah, into the livers of NOD-SCID mice. Do you know what that is? Is that NOD-SCID? Okay.
I know. No, I don't know.
All right. The tissue is composed of human hepatocytes and select non-parenchymal cells. Function of the 3D human tissue patches were seen via detection of albumin, alpha T, you know about this stuff, right, Zara? You know?
No, I don't know.
Oh, okay. Well, anyways, they were able to show that this tissue grafted, vascularized, it's working, and they're studying it now. So I think what you were saying about tissue, there may be a benefit to bioprinted tissue as far as how the tissue organizes itself with the extracellular matrix. You may want to look into the science, you know. So
you want to say that this is more advantage of bioprinting than making cells to make their own scaffold, or?
I think that statement is not necessarily true because it totally depends on the actual organ being reproduced. So what we don't know about the urethra, right?
Yes.
She knows a ton about the urethra in this case.
I can't. Oh no! I'm just throwing it out there that
Understood.
She was saying that the urethra had the phenotypes were after a couple weeks. It's a battle to try to have these cells maintain their complexity and allow it. So I don't know, but I know
like Organovo's thing is I can build a liver or I can build a kidney. In the case of the liver or the kidney, it doesn't matter the shape or really the structure of it. It can look totally not like what a normal kidney or liver looks like. The urethra is a very unique organ in shape and function.
Yes, right. If this is not, let's say if this is not stretchable enough, or if this is not tubular, so we are in trouble.
Right.
Yeah, and it may be cell dependent. You know, I mean, I know that you're talking. It's totally different because you're taking stem cells and turning them into those cells, whereas the liver tissue that's created that Organovo is printing is from a source of liver cells. So they haven't gone through that.
Yes, all these
metamorphosis. Yes, exactly. So
guys, this is pretty cool. This is you know, we're trying to hold conversations here, and there's this as we are live on YouTube, we have HealthSpan Discord. There's a few people from that group that are watching, so I've asked them to ask us some questions. One of the things that I find pretty intriguing is that, you know, here's the science, right? And then there's business, the science and business aspects of it. So sure, you know, Organovo has amazing science, right? Now you know, I'm not sure exactly what, wasn't able to read a lot about it today, but whether Keith Murphy is still on the board, or I didn't even read, so he's off.
No, he resigned because he's opening a whole new company that's going to do. He said in there that he's working on drugs that he's going to contract with Organovo to help develop drugs with the technology.
Okay, so you know, so basically, I kind of caught wind of that a little bit last minute, but you know, here's what we're faced with here as a company. It's like amazing science, and then there's business, and then how do you know, monetize this science? So there's, this Keith Murphy, he's a, he's out there, he's a revolutionary, he's out there trying to like wanting to get into everything, and now it's changed to a new guy, this guy Taylor, who's all about, picking and choosing the most profitable parts and staying focused and doing that. And it's kind of neat how, the science is, you feel like you can conquer the world, but, starting at certain places seems to be, the most profitable places, or maybe the path of least resistance. How do you, so our company has picked liver and kidney to go at, probably because liver disease in Asia, and especially China, is the most
number one.
Yeah, it's the number one problem over there. So where does what you're doing, why have you picked, you're talking about doing stuff with the urethra and stuff like that. Why did you pick that area? And what about the competition that's surrounding your field of study?
Mm-hmm. About your second question, which is competition, I don't know, because I'm not in industry, so I'm really not aware of the status of the commercialization or like these kind of issues surrounding that. But about why I choose, I cannot say why I choose, why it was proposed to me to do it, because this is very common happening of urethral stricture. Actually, this product is used to, how can I say, how it is used by urethral reconstructive surgeons to remove urethral stricture, which is very, very common issue in between male, and the occurrence is about 0.6%. And this is, you can say how much this is high. That's why this actually topic was proposed to me, to make this urethra to help patients who are suffering from urethral stricture. Yes, to help patients with this complication to solve their issue. Because so far the best tissue they are using for treating this condition is buccal mucosa, and this is not really easy option for surgeons to use, not for the surgeon and not for the patient. That's why this topic come to me to try to make it. Not only me, I mean other people who are trying to make. The main reason is the occurrence of urethral stricture in male somehow is high. And this is,
interesting.
This is the issue.
That's got to be painful. Yeah.
We got one guy that said, how soon will I be able to replace all my body parts with younger ones? And then he wants to know when he can maybe get a pair of wings. I told him it's going to be at least it's going to be at least a few years.
Yeah.
So we have. Are you familiar with RAAD Fest? The RAAD Fest? No. I'm gonna look that up while we're chit-chatting.
That's rad. It's rad. It's Rad Fest.
Rad Fest. Sounds interesting. Rad Fest. Cool. 2017. Take your place in the revolution against aging and death.
This is cool. Yeah, there's a whole construct around regenerative medicine, of which this conversation about stem cells and the conversation about bioprinting and CRISPR-Cas9 is all a part of it. There's no doubt.
RadFest is a global movement with the premier event taking place yearly in the U.S. We are excited to announce first international RadFest in Bangkok 2018. Pretty cool.
Bangkok. I would love to go.
That'd be cool, right?
Yeah, very cool.
Yeah, so another question I wanted to ask you. So, at school you're doing your master's degree and stuff like that, and Organovo has a lot of partnerships with different universities and stuff. How much of an impact are students and master's people going for their master's degrees? How much are you guys able to like push the science? Is it like, how intense is it? Like, can the next big invention come out from these partnerships? Or I mean, I'm sure it probably does, right? What's the feeling around that?
You mean how we as students contribute in pushing of science?
Yeah, exactly. So like so Organovo is saying, hey guys, here's our printers, here's our science. Now,
okay, but what I'm going to say is the company is the party which provide money, fund, and instrument, and the one who perform and introduce the knowledge or the science or make it practice and work is the students actually. So I mean,
yeah.
money comes from company, and science wise this is the students, either master student, PhD student. Means this is one of the hands of success in this field beside of the company.
That's awesome. That's gotta be cool just to be able to be a part of that, right? And get in the lab and work with all these really cool innovations and stuff. I'd like to take, and Sean, I don't know if you've heard this before, but I went to the University of South Carolina, and I'm close with the president of the universities, Harris Pastides, a really great guy. And then there was a guy this guy, Bill Kennedy, who runs a pharmaceutical company down in Florida. He's a billionaire. He's like a legit billionaire. And he's a big time booster. In fact, I think our pharmaceutical school is called the Kennedy Pharmaceutical School at University of South Carolina. So I'm interested in bringing, Matt and I are trying to bring Organovo to South Carolina to Organovo, and try to bring in that conversation, because we think it's a total win-win-win-win-win, with students being able to deal with some of the science, Bill Kennedy being able to maybe put some money up, or potentially even bringing in another partner that might be able to do some of the funding. So I don't know, some pretty cool stuff.
Well, what do you know? I mean, in that sense, Pat, Matt, what do you guys really know about Organovo's efforts to secure outside funding or money in general from people and or institutions in that sense? Because you and me, all of us, everybody listening, and the 25, 30 people on our email chain don't want to do this.
Right.
We haven't heard anything about what their efforts have been to get funding, right? Like outside of large institutional investors that you see on the public internet, as far as who holds shares in Organovo every quarter. But do you know anything about any of that? I mean, you're saying I'm trying to hook them up with this guy who's a billionaire, but I mean, I don't see anything. They're not releasing anything, I think, for good reason. But I mean, do you know anything else other than?
Well, I mean, the latest thing, Pat, can you pull up Organovo and NIH? We just received a grant.
Yeah, I saw that. 1.6 million.
Well, I mean, 1.6 million is, I mean, and when did that happen? I think it happened.
It was like a month ago, 2 months ago, right? But I mean, that's the first time we've ever had news of a grant, as far as I know.
No, there was other grants that were given early on, and I know that you could look in the quarterly filings as far as how much money was brought in by grants. They actually.
Yeah. I see all that. I mean, that's like 1.6 million over the 3 years of the term or something of the grant. I mean, 1.6 million in the terms, in my mind, in a company that's trying to innovate, isn't really a huge. I don't know who these people are. Chunk of change.
I know who Robin is.
I saw Robin one night.
Yeah, Robin's actually in this RAD conference, which is kind of cool.
Oh, cool. Yeah. Very cool.
I mean, I see like Gabor, right? Gabor, who used to be involved with Organovo, or one of the original founders of Organovo, is now doing meat printing, right?
Modern Meadow.
Right.
Yeah.
I mean, they just secured like 25 million or something from someone a couple months ago, and they're kind of an offshoot of all of this. And I've never seen that type of funding with Organovo. So,
I mean, Modern Meadow, it may be just a whole different, because their thought is they're actually bioprinting meat. So the conversation is around like ending world hunger. So you might have people that are behind world hunger in a way that aren't behind bioprinting tissue. That's the way
I get the
would say.
differences. I do. I understand that.
But I get the frustration too. You
But
know what I mean?
not frustration. I'm just more interested in how you guys. I appreciate you guys and your investment in this. I love it. Love where you're going, Pat, with your contacts in South Carolina. I feel like it'd be nice to know where Organovo was going more than where you guys are going in that sense. Like,
Look, I mean, I'm just trying to shake up the ground, and just.
I love that you're doing that, and that would be awesome.
And you're just learning and going. I mean, I want to just network people together with the goodwill that, hey, look, I've put these people together. I've shown my cousin's ability. My cousin's a great networker, and he's really passionate about this. I'd love to see him end up in a position at Organovo or something similar. Because I think that he has something to bring to that. And then as far as the university is concerned, it's just a win-win. Those people just need to understand. I'm not going to be able to be in the meeting and say, this is the science and this is this. I mean, I'm learning as we go along, but for me, being able to bring a Bill Kennedy and being able to bring, a Taylor into the room together and say, guys, let's talk it out. Oh, let's bring in Harris Pastides, who's the president of the university. Let's bring in someone from the Moore School of Business that's used to closing deals. Okay, great. I can bring all those people together with a phone call if it makes sense. So also, and then learning how to bring that deal together and help close it is also something that I want in my portfolio as far as like, hey, look, this is what I've been able to do is bring these people in that are like-minded and make some great things happen. Of course, you need the scientists. Of course, you need the money. Of course, you need the enthusiasm and the student participation and whatever else it takes. But like just to get the ball rolling is really important. And I would like to see Organovo or any companies that have their ears open to like where's the pulse, what's happening. I want to just, by doing these podcasts and by having conversations, we kind of just speak a little louder, and all of a sudden maybe we get noticed, and they go, "Wow, these guys are actually talking some good stuff."
Look, and listen, let me just say something to that too, because I've had, and when I said, "Hey, I understand the frustrations," because I've had some frustration about like marketing. I've wanted like, why can't this news get out there? And I expressed that on a recent phone call with Steve Kunzabo, who's the investor relations guy at Organovo. And, he said the return on investment is not there to go out to the general public. So the news, we're not getting the news. One, this is a different type of company to invest in because a lot of the pharmaceutical companies, they sign, anti-disclosure agreements when they contract. They don't want people knowing what they're doing. So we're kind of lucky that we get sometimes news of like Merck signing a multi-year deal with Organovo, which happened. So, or Astellas, or Bristol Myers Squibb. Some of these names, it's great to have them, but there's a whole host of other names that they don't tell us, and I don't think they're allowed to tell us, frankly. Yeah. And then he told me they don't invest dollars. The return on investment is not there to go out to the general public like me, the sales and marketing guy, or Pat, because it's really the scientific journals and publications and paying for the studies and putting them in journals that we never even see, Yeah. So,
I mean, you know what, for me though, and beyond all of that, and I don't know if Steve, who was on the call with us, it's almost like, Zara, how did you get started? Where did you get your inspiration from? Well, the more and more that kids understand what's happening in, like CRISPR and things like that, like that's all of a sudden becoming a buzzword now. I hear it. I don't hear it often, but I hear it more often than I used to. You hear Bitcoin, you hear Ethereum, you hear, digital currency quite a bit. Like I was supposed to be mining digital currency 10 years ago when it was $1. I would be like a multi-billion. I would say, hey, let's go. Let's start our own little thing. I mean, that's the kind of money that's in it. And this is stuff that is not about money, but it's about saving lives. So why shouldn't it be talked about? Why shouldn't this conversation be like out there for young kids to pick up on? For even old, like Matt is, you know, I don't know, 28 years old. How old are you, Matt? No, Matt's, you know, he's getting up there, but yet he's now all of a sudden his interest is in bioprinting and CRISPR and regenerative medicine. He's talking about this. He's living every day. He's in there. He's so excited. He calls me in the midday. Oh my God, you can't believe it! I just read this thing. I mean, he's like a little kid right now because.
Yeah. Did you see? I don't know, Sean. Did you see? Do you know a guy named Bas Rutten? You ever hear that name?
I think wasn't he on one of your little webcasts or something?
So he's a guy that is like a world champion MMA fighter. You just saw the fight 2 nights ago, right? Or you heard about that? Yeah, so the grandfather of that stuff is Bas Rutten. Pull him up, you go nuts seeing him. I mean, he was a beast from the Netherlands. Well, he has this volumetric muscle loss in his arm, and he thinks it was like a nerve block. He went to Panama for a stem cell transfusion because you can't get that sort of procedure in the United States. Right now, I believe in Texas, they're the first state that allows you can bank your stem cells wherever in any state, but actually having stem cell therapies, you're not allowed because they're worried about people like doing the wrong thing with it, et cetera. This is what I think. And I read that Texas was the first state to allow like stem cell procedures for patients that are in dire need, though. It's like really, can you pull this up, Pat? I mean, I want to make sure I'm right. Like Texas first state stem cell, you know.
Texas.
That kind of falls in line with that FDA release that came out today a little bit.
What release?
So the head of the FDA did that big release today on basically saying the FDA is putting out new regulations towards bad actors in stem cell and tissue regeneration industry to make sure that the appropriate organizations are performing legal and or safe processes within their business and not trying to dupe and perform unsafe procedures.
Yeah, absolutely. I think there's a lot of people in the the health and beauty type of market that will try to sell therapies for people to look younger, feel younger. That happens all the time. But really, when you get into like macular degeneration, or I think it's going to benefit people that before they have a knee replacement, you could, as long as you have enough cartilage in behind your kneecap, you could inject your own. This is just my theory, that you could inject your own stem cells if you bank them, and it will grow inside your kneecap, and you'll have essentially a brand new knee. You know, I mean, that's what I think can happen. When was this article, Pat? Can you see a date on that?
This article was May 30th, 2017.
Okay, so
pretty
couple
interesting.
months.
So let me just get to some of these questions that the audience over here has. We got a few questions here. One guy said, let's see. Haynes says, about 2 years ago, I read an article that claimed there was success in 3D printing blood vessels and that this could lead to 3D printing the liver. Any thoughts? Well, there has been some. I know that Organovo was working on some blood vessel stuff, right?
Yeah,
really talk about it too much.
Yeah, I mean, if you pull up Organovo's website and you look at the history of it, I'll show you. It's pretty cool. I mean, you'd think, hey, there's a blood vessel, but it's not. It has to do with I think it has to do with the pressure. Go to the science and technology and the history or whatever. That's Gabor, I guess. No, you got to go to the origins. Yeah, origins. You see that little tubular structure over there? That's the first bioprinted tubular structure ever. This is Gabor Forgax at University of Missouri, Columbia. These are the foundational bioprinted structures that he created. He was working on chicks, and then saw that the cells would kind of coalesce around each other, and said, I wonder if I can make that happen. And he printed, and So that tubular structure, though, is just strictly tubular, and they have foundational patents on some of this stuff. But to your question, Sean, I think the problem, and they've done all kinds of things. One study in Pennsylvania, I believe, they used very, very fine sugar, strands of sugar from confectioners' machines, and then coated the cells around it, created the structure around it, and then with warm water, they dissolved to try to get that fine vascular structure. And right now, they're just not there. That we are definitely, from that question from that guy, we're I don't know. I mean, we could be 10 years away, maybe less, 7 years away from the vascularization problem. Although with some of the stuff that's going on now with self-assembly, there is the potential for a vascularization structure that's not bioprinted. So I don't know. It might be that there's some kind of marriage between the two that could, because I saw something recently on vascularization and cell structure, but it's not there yet in my opinion, for your guy that's asking the question.
Mm-hmm.
But they can do patches that'll bridge the gap. That's where we're at right now, is patches.
Right. ES says Organovo is great. Looking forward to interventions that won't require surgery, especially optimistic about rejuvenation, biotech, and our ability to get aging under medical control within our lifetime. He also thanks us for bringing the conversation regarding regenerative medicine. Haynes said it's worth nothing now, but it has a good trajectory. I guess he's talking about the stock.
Yeah, that's why I'm buying now.
HealthSpan Hub got your back. HealthSpan Hub's got your back. Right on. Glad you guys got our back. Medicine and tech in general never progress at the pace it's progressing at right now. Do you find that true, Zara? How do you, since you got into you're now a master's student, so you must have been in school for quite some time. What's the energy like when you first got in? Is it a different buzz that's happening in the lab, or around just your friends and people that what's the overall feeling in the science today?
Can you ask your question in another way, because I didn't understand much?
So you've been in school for quite some time, right? So like at least 6 years, right? So at the very beginning when you got into school compared to now, what is the overall like energy? Is there more talk about these sciences? Is there more people getting involved? Is there more excitement around this science than there was when you first got into it?
Of course. When you get more knowledge about the field that you are working, of course you become more interested. You will see more progress. You will be having more energy for doing that. I mean, of course you are not like the first day you enter to this science, to that field. As much as you go through, talk with people, know about information, and you see the result of your not only your work. I mean, let's say in my field in tissue engineering, maybe I'm on the way of making my own, but by my own eyes in the hospital, I can see that the other people making, using, I see patient is healing. So this is, I think, within these years, by seeing these results and hold these events, of course, I'm not the same as the 6 years back. I mean, more interested, more passionate.
Yeah, so like for instance, like in the early 80s for me when I was getting into computer. Really,
oh my God, early 80s.
The late 70s, really. What am I thinking?
Jeez.
So it was like the late 70s getting involved with computers. There wasn't a lot of people in like the computer lab or people hanging around talking about computers.
Or investing.
There was no computer lab.
There was no computer lab, really, right? And then all of a sudden, 10 years later, there's huge. So like I want to draw the parallel between science, like this regenerative medicine that I didn't really hear too much about 10 years ago, or even 6 years, 7 years ago.
But yes, now much is more people are more talking about that.
Yeah. So speak a little bit more about that. Like, is there any? I'd like to hear something specific that hit you, that like from 6 years ago, or maybe it could have been 10 years ago when you were like, hey, I want to go to school for this. Was there anything that strikes you as being like, wow, this is catching on? Like people are catching the groove here with this regenerative medicine.
Yes.
About?
I think the first time I heard something about like regenerative medicine or tissue engineering, which for me it looks like a science fiction more than a reality, was Dr. Prof. Atala's speech on YouTube. I saw some video on YouTube that he was talking about the organs that they are printing, they are matter. I'm just saying, for me at the beginning it was look like a science fiction. I thought, is it really real? Then I think from that point, that was the thing that hit me to go and read and become interested, and at the end to be involved in this field.
Was that Dr. Atlas? Is that what you said? Atlas?
Yes. Prof. Atala.
Okay. Oh, okay. Interesting.
From Wake Forest, Atala.
Yes.
He did a TED talk where he printed the shape of a liver on
bladder.
Yeah.
Oh well, yeah. He did the bladder, and actually was the first. Pull it up. He did an amazing thing, Dr. Atala, Atala. He printed. He grew the cells around a scaffold and saved a little girl's life, basically. Pull it up. It's cool.
If I'm not mistaken, they are practicing some kind of bioprinting for the one that I said, for ureter, in their lab also, like currently in 2016.
Well, yeah, maybe we want to take a look at that too. After, you okay there, buddy? You able to get the?
Yeah. What do you think this is? Like it just you could just. Huh?
I mean,
I didn't spend enough time in a computer lab, apparently.
Okay, there it is. That's part of it. You're it's not a TED talk though. Well, you got to go to.
Anthony Atala asks, can we grow organs instead of transplanting them? His lab at the Wake Forest Institute for Regenerative Medicine is doing just that, engineering over 30 tissues and whole organs.
Yes, tissue engineering. And you see the scaffold over there, that picture? If you pull that picture up. So Pat, that's the scaffold. And then what they're doing is they're putting, seeding it with cells.
Yes.
So they're seeding it with cells, and they put it in a like a baker, it's got the proper temperature for the cells to grow and proliferate, and then it creates like a mold, and then they're able to use that mold to then use the tissue, and they actually helped a little girl with that very method. Is that what you understand? Yeah.
Yes, right. But at the beginning, it looked like a fiction for me. That how come it? I mean, because everything was really in a gross structure and like a tissue.
Yeah.
So it at the beginning it was unbelievable for me. Then yes.
And what I was talking about was a TED talk that he did where he talked about the future of bioprinting, which got a couple million hits, like it's on there. And at the end while he was talking, there was a 3D printer in the background printing, and then he had medical gloves on, and he showed everybody this thing that was in the shape of a liver or a kidney or something, but it was printed out of it was rubber.
But it wasn't what I mean? It wasn't functioning.
But it created the buzz around bioprinting, though. So there's a real benefit to it. You know what I mean?
Yep.
So you watch it, you'll think, oh my God, he printed a, but it's
human organ. Human organ in his hand. Yes.
But the greatest
that
part.
Was right around. I'm sorry, Pat. Go ahead.
No, go for it.
That was right around the time that Organovo got caught up in the 3D printing craziness that launched it to crazy places.
Yeah.
Whereas it's really a bioprinting company.
That's right.
I've always considered that Wake Forest fella, Atala, and the Wake Forest group, because they hold a significant number of patents as well at the university. Almost like, because we don't talk to them, we don't like correspond with them too much. I almost feel like they're in competition with Organovo, despite the fact they're not a public company.
Right.
I felt that way.
Yeah, and I mean, competition, seeds greatness. I mean, I think that
Sure.
competition's always a great thing to happen. Um, the funny thing is just kind of what I was trying to get at. You watch that thing by Attila, and it inspired you. So conversations all of a sudden have this young, intelligent woman getting you know, into the study of regenerative medicine. That's awesome. So that in itself is why marketing is a good thing, because you can consider Attila's, you know, speech or TED talk marketing, really, marketing by someone. They're selling something, they're selling advertising or whatever, and that is a really great thing. So that's why I think marketing, and there has to be more conversations and more people involved in this stuff. So
Well,
so
Pat, hold on
Paulo
a sec.
says,
Right before you get to Paul, I really recommend that
Paulo,
people go. All right, Paolo. It's Paul, but it's not Spanish.
it's
Okay,
Italian,
Paolo is Paul.
Italiano,
All
obviously.
right, so hold on one second.
Fidago.
People have to take advantage of TED Talks. If you want to learn about, like you mentioned, Gabor Forgax and his son actually are doing the modern meadow. That's a TED Talk. You can learn about where they're going. Not only that, they're going to print leather for the car interiors, like that sort of stuff. So,
Right.
to not deal with animal stuff. But TED Talks are an amazing opportunity. Anybody that's listening to this that watches the Atala talk, which is years ago, that's like a 5 year ago conversation, easy, maybe 6. That was the beginning of the hype, but the promise is there. You look at Organovo and you see the tissues that they're constructing with bioprinting, and you look at the pharmaceutical companies that are contracting with them, that their toxicologists are looking at these studies and they are saying, oh my gosh, this is wonderful. I mean, it's more predictive. And not only that, they're creating, and no one else is doing this, disease tissue. So they're taking liver tissue and they're creating a disease state. So for NASH, that's what's causing over 50% of this last quarter's contracts were for the disease tissue, because they're providing disease tissue services where they can test drugs for efficacy. And this is extremely powerful. So, but the TED Talks, I highly recommend it. What did Paulo have to ask?
So, no, actually, I absolutely agree with you, and I think it's a great thing that Organovo is going into the disease modeling, because that's where they're finding most of their profit, and going into the profitable areas is a wise business move. So you always have that science versus business, and I think Taylor is one of these guys that is able to, give the right little bit of business where it needs to be business, and the right little strokes of science where it needs to be science. So Paolo Paolo Fidago must be Italiano. My cousin and I are
That's not Italian.
Fidago's
Is
not
it?
Italian? I think so. I mean,
You
our
think?
mothers are
I think
Italian,
it's
so
Spanish.
that we're actually Italiano, a bit half. I'm Irish. I'm not sure what my Uncle Wally was. Um,
Better.
so medicine. So Paulo says medicine and tech in general never progressed at the pace it's been progressing right now.
Boom!
Okay, so he says exponential, some might say, which is awesome, and that's why I wanted to know, like, what was your feeling. It was really cool that you mentioned that guy, because we, we're kind of in that.
We know what you're talking about.
We're in
Yeah,
that
we know what you're
same.
talking
We're in
about.
that same
Yeah.
realm. He says it's only going to accelerate more. The law of acceleration returns.
Hey, do me a favor. Pull this up for Paulo. Look up, Pat, the number of publications for stem cell just reached like 300,000. There's a graph. The number of cited publications or something like that. It's just a graph of the publications on stem cell.
Yeah, this is good.
Did you find it?
I did, I think.
Graph on publications, or graph on scientific STEM trends and perspective. Oh, you got to write like graph on, you know what I mean? Like Google graph of stem cell publication mentions or publications. Selvia published. And I just saw this the other day. Number of publications were up to, no, but it's like 300,000 now. I don't know when this is. Anyways, the point is that more and more, that's backwards. That looks, but I would have to find it for you. The point is that, like you're saying, there is an explosion of conversation around regenerative medicine. You look at CRISPR, how much CRISPR is done, which is gene editing, stem cells, which, how many different types of stem cells are there? Like 5, is that correct? There's pluripotent, right?
Are you talking to me?
Yeah, Zara, yeah.
Yes, we have embryonic stem cells.
Umbilical, yes.
Embryonic stem cells, and
embryonic,
like
yeah.
five, six different stem cells, like adipose-derived stem cells, which is
Adipose.
mesenchymal stem cells, bone marrow-derived stem cells, urine-derived stem cells, the one which is derived from Wharton jelly.
Yeah.
This is what at the moment I remember.
You know, and I think some of the people that listen to stem cells, they still think that it's embryonic, like it's got some sort of danger to a baby. And we are way, way past that. No babies get hurt from this. This is the cord after birth. That's the real young blood. That's where, yeah.
But it is still not embryonic stem cell. The one which is, if I'm not mistaken, the one which is get from the cord is not embryonic. This is somehow adult stem cell.
Yeah, but we don't use the embryonic, right? We don't really use the embryonic.
No, we don't. First, we have ethical issues in using that, because from where you are going to get that embryonic stem cell? This is from fetus, from womb. But second, this is, there is a, since they have their pluripotency, this is much chance of developing tumors after you use them. But since they reach to the stage of adult, means embryonic stem cells, any kind of adult stem cells, bone marrow, urine, fat, the chance of making tumor is less, and they are not ethical. I mean, there is no ethical issues for having them. And
that method was created to create fat cells into stem cells by a doctor in Japan, right? Yamakana, is that correct?
The fat, you mean?
Changing a fat cell to a stem cell that you could then use.
No, we don't change fat. Stem cells are everywhere in body. You just have to have the knowledge to isolate them. Stem cells are in between fat cells, which is known as in a portion which is known as a stromal vascular fraction. Throughout the process, we isolate those cells and throw the rest. The rest is like blood cells, some immune cells, some red blood cells, fat. Yes, we throw them out. The one which is left is stem cells. So we don't convert fat to stem cells. Stem cells are there. It's like bone marrow. Stem cells are in bone marrow. So you have to have knowledge to how to isolate it.
Okay, so once they're isolated, say you have stem cells, then to change them into a different cell, the one you want, if you want to change it into a smooth muscle cell, or a certain cell that's in the urethra, right? Yes, there's a very, very specific process to change that cell to that specific type. And now maybe it's that method that was developed by a guy who won the Nobel Prize in medicine, I think, right?
No, I think if not, I'm not mistaken, you are talking about induced pluripotent stem cells, which is.
I think I am. Yes, that's a different thing.
Induced pluripotent stem cells are adult cells that I think that was a team in Japan, the person that you already mentioned the name, but I don't know now at the moment. But I think they used adult cells which doesn't have pluripotency. They don't have this flexibility to change to other cells. Then they transfer it back to stem cell, which is called induced pluripotent stem cells, by introducing some genes, if I'm not mistaken. So we have stem cells now, which is made from adult cells, means this throughout a reverse process. So they have it in their hand. Now they can convert it to any other cell type.
That's pretty amazing.
So, yeah. In a way, you isolate the stem cells from the patient or from the person. In another hand, you can have the adult cells, convert them back to stem cells, then use it for whatever other purposes you want to use. This is induced pluripotent stem cells.
Now, I
Now, do
mean,
we
yeah,
know?
go ahead, Pat.
I don't butt in here, but just someone asked about extended pluripotent stem cells. Extended pluripotent stem cells. Has anybody heard about that?
I have not.
No. So just
Me
another.
too.
We have some of the guys. We have 13 people live watching. Just
Hey,
asking
what's
normal.
up?
So everybody say hello. Our friend Paulo is he's Portuguese. So
Uh-huh.
I was close.
Not as close as me, I think.
Where'd you
Yeah.
say? Where'd you
I
say?
said like Spanish and Portugal and Spain. I mean, you're
Is it?
taking the
Okay.
trip to Italy? You're talking Italy?
Well,
I
it's
think
right there, buddy. Portugal's not
Portuguese
too
is
far away.
closer to
Okay.
Spain than yeah.
So
But
anyway,
what's up, Paolo? Yeah, like
so
yeah.
no, we have so Haynes did 17. He researched this. He said 17,000 research papers matched on free search.
Yeah.
So apparently, we do have a little bit of interest growing because 10 years ago, I don't think you had a whole lot. Or 5 years ago, you didn't have a whole lot. And I do think it's probably hitting this like trajectory right now within this like last year. I mean, I hear a lot of people talking about regenerative medicine, and you know, it's it seems like hopefully, if the FDA kind of loosens up a little bit and we start to be able to push some of these things through.
What? That's a great point. The FDA, if you look at the Cures Act that came out recently, I mean, it was actually signed into law last year. Pull it up, the 21st Century Cures Act. Uh, you know what I mean? And just do 20th Century Cures Act and tissue therapies, and we'll see the language of it. There is very clear language in the
Say
law.
it again. What do you want me to look at?
Look at the Cures Act and cell and tissue-based therapies. The Cures Act, Cures Act,
I got that.
and cell and tissue-based therapies. And then hit enter. Come on. I want to see it.
At all. Taking dendritic cells into medicine, pitfalls, or engineering principles. I don't
You
clinical.
don't find is it the Cures Act? Put in the Cures
I
Act and
put
see
in
what
the
comes up.
Cures Act and
And what did
cell
it
and
say?
tissue-based therapy. I got a million
Just
articles, like.
all right. Anyways, you're going to have to do the research on your own. But I guarantee you, because I looked through the Cures Act and it's like 500 pages, and I found it on like page 320 or something like that, where it said that there is going to be a streamlining of processes for cell and tissue-based therapies. This was actually a press release that they were really happy about from Organovo about a year ago. So there's to your point of, hey, what is the FDA doing? First, they're like you just said, they're concerned about stem cells. They're very concerned about CRISPR-Cas9 because gene editing is crazy. I mean, it could be like super powerful, and it also could wipe out Down syndrome like that, like gone, you know? So, yeah, yeah.
Yeah,
So
yeah.
it's like, but then there's a push for the U.S. government to do more in stem cell and tissue therapies. I guarantee you.
Yeah, so there's something that ES over here has said. Eram's law. Have you guys heard of that before? So Eram's
Hey,
law is the observation that drug discovery is becoming slower and more expensive over time, despite improvements in technology such as high throughput screening biotechnology, combinatorial chemistry, and computational drug design. A trend first observed in the 1980s, the cost of developing a new drug roughly doubles every 9 years. In order to highlight the contrast with the exponential advancements of other forms of technology, such as transitions over time, the law was deliberately spelled as Moore's law spelled backwards. Oh, that's cool.
hey, Pat, can I
Yes.
share my screen?
I don't believe you can.
Can I send you a link that you can share?
Yeah, do that. You can actually just do it in this thing. If you look to your right, you can see a little messaging thing. Yeah, hit that little.
Look at that. Hit it.
There
Click
we
on
go.
that.
Yeah.
Click on that.
Cool.
And then scroll down to the bolded section titled Efficient Regulation.
Hold up here. Something happened with me. Okay.
And in the third paragraph or fourth paragraph of the section titled Efficient Regulation, you'll see, this came out today. This is what I was referencing earlier. You'll see their referencing of the Cures Act.
Where do you want me to go here? Third paragraph?
Go down to the Efficient Regulation section. This is the FDA. There you go, right there. It's third paragraph right there. And this references the Cures Act and where in the fall they're saying new policy is going into effect. So whereas the Cures Act was established, what Matt said, about a year ago or so, there's actual movement and
I'll read this out. I'll read this out for the people. The FDA will advance the new framework this fall. The comprehensive policy will establish clearer lines around when these regenerative medicine products have sufficiently complexity to fall under the agency's current authority, and then define an efficient process for how these products should be evaluated for safety and effectiveness. The policies will be set forth in a series of guidance documents that are the result of public process we have held in recent years. The new policy will build upon the agency's current risk-based flexibility regulatory framework. It will also serve to implement provisions of the 21st Century Cures Act related to the regenerative medicine. The FDA has already held public meetings to inform its thinking in these areas, so much of the agency's approach is already part of the public record. We'll continue to work with the industry and the scientific community to perfect the process for bringing safe and effective treatments to patients. That's awesome. And somebody else chimed in here about, yes.
You can share a screen and then you can make it bigger if you want.
Oh, that's true. Someone said throw Trump at the FDA. Well, it seems like from this paragraph we don't even have to do that, but I believe that Trump in his inauguration thing had said something about trying to fast track things. So who knows? Maybe he'll be good in that regard.
Or not.
Or not. We'll see. You never know. But let's see.
Hey, do another one. Just President. All right. See, I'm reading one right here. President Obama signs 21st Century Cures Act, creates accelerated approval pathway for regenerative medicine.
So the act gets signed, right? Now the FDA, after 8 months, is finally getting word.
Well, they don't know how to implement. You know what I mean? It's a gigantic bureaucracy. So the point is, accelerated pathway for tissue and cell-based therapy. Like, let's go. You know,
impressive that after the Cures Act that Obama signed a year ago, we're actually now 8 months in and seeing the FDA doing something. I'm impressed in the 8-month timeline that's taking place. Like,
yeah,
under any other thing, it would have taken years and years and years. So maybe we're actually doing something.
I think we're on the right track.
And this came out today.
Today.
Wow, I'm gonna go back to it. Sorry.
It's a long article. Yeah, that's just a subset of. Send that to Matt too. I'm sure he'd like to read it.
Yeah, no, that's great. I appreciate it. I got you. I'll read this. This sounds great. Yeah, I've been keeping, I've actually been kind of keeping, like, I haven't really read much on how much they're trying to stop people from using stem cell therapy, because I think there's a big backlash that is just crippling the use of stem cell therapy for people. I mean, I posted something today. I'm like, we were drowning in high fructose corn syrup because of the Corn Act from the 90s, and you look at diabetes and pediatric diabetes and in this country, and it's amazing what we're approved to eat, like a million Oreos or whatever, but they won't let you take cells that could be taken from your own body and just kind of moved into cells that could help you, and and replace them. I mean, I find that absolutely like
that's a good point, Matt.
It's backwards, you know. They'll let you eat plastic, but you know, you can't.
Much worse than plastic.
Yeah, like I mean, I just heard, you know, microwave popcorn is the worst thing for you. I just found out the other day, you know.
And then there's cigarettes.
Oh yeah, then there's cigarettes. They're approved, right? But let me take my own stem cells to try to help my bum knee or whatever, or Pat's bum knee, I should say. So.
Yeah, you know, it's crazy that as a human being that you don't have the rights to do whatever you want to do.
Preach.
I mean, but you know, but I'm not saying like you know, as long as you don't, you know, impede on other people's whatever, you know, all that kind of stuff. But if you're dying and you were like, hey, look, I'm in so much pain, I want this therapy. I want to give this guy, I want to give my cousin who's came up with this cream, I want to be able to eat it, you know. Like,
yeah.
I don't get
Well,
this.
no, you look at people who die from liver disease and are on the organ transplant list every year. It's 30,000 people a year are dying waiting for a liver transplant. And you know, that's why this liver patch is important from Organovo. It actually can bridge somebody for like 2 years while they're on the waiting list. And they found that when they patched the liver tissue on there, it actually had a curative effect on the tissue next to the patch. Like it actually has a potential curative effect in a way that they're trying to understand. But yeah, it's tough. You got to try to. You got to keep pushing.
So Haynes says cigarettes are delicious and screw the FDA. Come do your research in South Africa, he says. Paulo, our Portuguese friend, says, what's your opinion on tissue nano-transfection, TNT? I've heard a little bit about that, but these guys know more.
That's like an AC/DC song, I think, right? That's what I think when I hear TNT. Dynamite.
Yeah.
I don't know any. I mean, nano. That's a whole nother thing. Nanoparticles. I mean, you talk to some of the regenerative medicine people. I think nano is awesome. There's the potential for little pre-programmed nanoparticles to go into your bloodstream and repair your cells inside of you, and then just pass through, and you have healthy cells on the inside, from the inside out. So.
Wow.
That's potential therapies, but I don't know what he's talking about. That was specific.
He's talking about. We're going to share the screen. This is
go ahead.
Cool.
Paolo.
Okay. Paolo comes up with this thing here. This miracle medical chip could one day heal almost anything.
Yeah, this is a New York Post, so be careful. Hold on, let's see what it's saying.
Imagine a future where car crash victims or Alzheimer's patients could be healed with the touch of a button. Scientists may have taken the first step to making that a reality. Researchers at Ohio State University have developed a small dime-sized silicone device known as tissue nano-transfection that uses nanotechnology to program a person's cells. To work, a doctor merely needs to place the chip on a person's wound, and the device sends an electronic pulse that converts living cells into whatever cells the body needs them to be. The pulse opens a small window into the cell that allows the chip to resend a new genetic code, and the whole process takes less than a second.
Okay. Now hold on. This is the most important part. It's the device repaired injured mice legs, and it was published in Nature Nanotechnology. So I know that Nature is a good publication, but I'm not sure about Nature Nanotechnology. Yeah, to push a button and cure yourself, this is a little bit.
This guy looks pretty smart, though.
Yeah, because he's wearing a glove. Come on.
I don't know. Just his eyes look like he's smart. It's like the way he's got smart eyes, and he's staring us down like he's got camera like right at you. Like
I don't know.
I'm a smart dude. That's what he's saying. And I got a chip that'll cure you.
Well, everything's possible. You got to read up on it, though. I mean, the science is where it's at, you know. So
it's interesting.
to Paulo, I would say don't believe everything you read, but read a lot more on this one. Keep your mind open on that.
Let's see what else. He says, ES says TNT is super exciting. Touch the skin with the A chip, press a button, inject DNA, RNA, transform the cell into any other type in a non-invasive way. Haynes says, at which point does bioprinting overlap with regular tech to the extent that nanoparticles are considered biotech?
I don't get it. I mean,
He's saying
nanoparticles.
when do they kind of converge, or when do they overlap each other?
Well, I think that there's the potential for a convergence with cell therapies, like stems or like CRISPR-Cas9 type stuff with potential genetic diseases, especially right now with the genetic, like the way they do it, they'll inject vectored cells into the bloodstream, hoping that they get to the area that needs the repair. But with bioprinted tissue that could be like grafted onto an area, if there's a disorder that could be benefited from vectored cells that can go in and repair or change the genetic profile of a disorder, they could be delivered right to the site that needs it. And some of that was kind of touched on in the conversation that Dr. Eric David, he's the co-founder of Organovo. He did a genetic, it's a recent YouTube. Go to YouTube, Organovo, Eric David, and anybody can find it. That's the closest I could come up with.
When was the last time anybody heard from Eric David?
I sent a message to him not too long ago. I asked him if he was a runner because he's like really thin. Pull him up, Patrick. Pull up Eric David. And he's like a thin guy, so I asked him if he was a runner because I'm a runner, right? Pat wants to be a runner.
Yeah.
And yeah, we're about to run the
Really?
Baltimore Marathon on October 21st.
You don't have to be thin to be a runner.
Are you?
I'd rather
Yeah,
run my mouth.
Well, the reason I'm bringing that up is because Pat's got an integrity problem, which is that he said that he's going to run and he actually signed up for the Baltimore Marathon. And that's coming up in 8 weeks. And he's maybe run like 8 miles total in training. So I'm just saying.
I'll be all right. Don't worry about it.
You should try the 1 mile, the Fifth Avenue mile or whatever. It's just a mile.
I've been running on a treadmill too, so I'm sure when I jump onto the pavement, it's going to be a little bit different. But
why don't you get you can get one of those nano chips, and every mile you can just hit it to make you feel like you're fresh again. Yeah.
Well, let's see, at least one more. What do we got here? Let's get back to the focus here. Let's see. I have a legit question. If we kept replacing the body's old organs with newer ones, would the brain live longer?
Well, why not inject some stem cells into the brain? Is what I'm saying. You're gonna rework all the cells.
Well, that's the mind-body thing. I mean, I have in my real life, I'm a animator.
You're gonna get off topic.
No, I'm not. What are you talking about? Why are you always jumping the gun?
Sounded like a long story. I'm just saying.
No.
Back in 1980, I was
I
like,
have our characters as beings, and their bodies and their heads are separated from each other, meaning that their body actually controlled them rather than their brain. So it's like this mind-body thing, and I think that's what this person's talking about is like. If you start replacing the body parts,
what are you gonna do with the brain? Yeah.
Boy, is your brain getting younger.
Well,
your brain definitely doesn't have to work, but it depends. Your body could actually control your brain.
Pull up one more thing. Do injecting stem cells in the brain and see what comes up.
Hey, Matt, you sent
Yeah.
Eric an email and just asked him if he was a runner?
Yeah. And he said he wasn't, but his wife was. Hey, man,
Oh,
did you listen to that podcast of his? He is great. He quotes like
I
Hemingway and
I
stuff
didn't
like
know he
that.
had a podcast. I know that in past presentations he's done a fantastic job, and I've been impressed with him, but I hadn't heard much from him lately.
Oh, no. But I'll send you the link to his podcast. You got to watch the whole thing. He curses at the end of it. It's really funny. It's good.
Is it a regular thing?
What's this? Oh no, don't say that people with a stroke are walking again. What?
Daily News. I don't know. You told me to bring it up. I mean, do you have a follow-up talking about a
Well, I
long
mean, is
story
that
that went nowhere?
Oh,
What do you
okay. I
have?
guess I guess people having a stroke in a wheelchair, getting up and walking again is just that's nothing.
Okay, well, what
No.
do you got?
Not only that, they did well, you just took the first thing that you saw, right? So there's a whole host of things that came up. Go back to your Google search. I know there was a study where they took stem cells and they had people that had, because I listened to the podcast because I was waiting. I was on, you ever been on like Eastern time and you're listening to something that was on Pacific time? So it was like a couple hours
Stem
early.
cells have been injecting into the brain.
Yeah, look, they're trying to do it for Parkinson's disease, stem cell, safe for stroke, chronic stroke patients. Maybe that one, injecting modified human adult stem cell. It helped them. I'm telling you, they studied patients that didn't have, they had already had a stroke like 2 years before, so they weren't expected to have any movement from their, um, their state of no gains they expected. And they injected stem cells into their brains and they had marked improvement. So to your question about your brains, our brains, wrap it up. Yeah, I got to wrap it up in my family because I got to put the kids to bed. But
Well, yeah,
we got,
they're
yeah,
saying we still have 10 people on here, and that's what they wanted to know. It's like a little philosophical thing, because there is that mind-body. We don't know a whole lot about the mind, right? I mean, we're learning more, but we still only, what do they say? We only know 10 to 15% of,
something like that.
yeah. Um, so Paolo says TNT is on medicine.osu.edu. Haynes says, what do you think of Dr. Shea Afrati research on hyperbaric oxygen therapy as a catalyst for omnipotent stem cell generation? I know a
I don't
lot. I
know.
mean, I've heard about the hyperbolic chamber stuff.
Tell him to join us on the next show and tell
Haynes,
us what he
why
knows.
don't you pop in on the
Yeah.
next show, and you can, uh, be on the, what do we call this?
Send
The panel? We call
send
this the
Pat
panel.
send Pat your email and he'll invite you to the
Okay, we got a
next one.
couple more things to see. Can you please try to find that page? It seems I cannot post links here. Oh, she wants me to get, or he wants me to go into this thing. Okay. Let me see
Yeah,
if I can.
I gotta run. All right,
All right,
guys.
jump out, Matt. We'll
Peace. I'll see
continue
y'all later.
later,
Good to
buddy.
meet y'all. All right.
So
Bye bye.
he wants me to get into this. Let's see. Where was that? EDU thing. I don't know where it's at. But yeah, there's so much cool stuff. Here you go. Medicine.edu. Let's see what that has to offer. I mean, creating the future of medicine through innovation in education, research, and collaborative care. And it's under TNT, I guess. Tissue nano-transfection. Breakthrough device heals organs with a single touch. What? That's pretty interesting. Living skin cells. Let me blow this up for you guys a bit. Wow. Sorry. Go back down a little bit. Sorry. Okay. Injects Genesis code into skin, turning this. What kind of
How do you
stuff,
how do you inject genetic code into skin cells?
Sarah?
Uh, I don't know. This is not the area of my expertise.
Yeah, it just sounds crazy.
uh,
Gene therapy, it's been around for some time.
Maybe they use some, if they want to do gene editing, maybe they use some primers to integrate with the DNA. I mean, they can design DNA based on their own interest and introduce it to the cell through electrospinning, if I'm not mistaken. So the DNA can integrate, and after several times of duplication, it can integrate inside the DNA. So this is how they edit genes, or they can inject genes, if this is what you asked.
Because this is interesting. Because the technique uses patients' own cells and does not rely on medication, researchers expect it to be approved for human trials within a year. Wow. Wow. That's pretty wild, huh? So,
Expectations.
to,
Ambitious expectations.
I'd like to see this thing happen. You know,
All
how
right, y'all.
often,
I gotta roll.
okay. All right, brother. All right, Sean.
See
Be
you.
well.
Zara, nice to meet you.
Zara, I guess we'll hang on here for a little bit until we get through these questions, just to respect the other people here. Let's see what we got. That's really some interesting stuff. Let me close that there, and then we're going to go to. Okay, Haynes says, "What do you think of Dr. Shea Efrades?" Don't know too much about that. I acknowledge Matt's joke. Let's see. Omnipotent stem cells in the bloodstream find and fix probiotic areas automatically. Do you know anything about that? Omnipotent stem cells in the bloodstream? Find and fix.
No.
No. Okay. Let's see. Yeah, when you see there was an experiment done. Let's see. No, just a peasant CRISPR. I just watch a lot of TEDx videos. So Haynes is on that TED Talk stuff. TED Talks. That's where you actually got involved with
Yes.
your study, right, and your inspiration. That's really great. So what else has inspired you, and like where? So you've been put into, you've been directed to study urethra stuff and along those lines. Where do you eventually want to go after school? Like what is the pathway into the professional world for you?
For your first question that you asked me, what made me inspire more? I mean, other than that TED Talk, when I see patients receiving, I just see few trials that they are receiving engineered construct and they become fine, they cure. So I think this is the best thing that for me can inspire me to go further. But in future, for now, this is like not really clear where I'm going to go, but I don't know. I will just continue whatever research I'm doing to see what's coming after.
That's great. And
If we can make it in clinical state or not. So this is maybe my aim in coming for 5 years.
Mm-hmm. Now, do you typically work within groups?
Yes.
Okay. And I did, different teams. Are there many different teams within your lab, within the school?
We are a tissue engineering team, and working together with different surgical teams like neurosurgery team, brain surgery team, heart surgery team. So these are the ones that we cooperate. So tissue engineering team with different surgeons from different specialties. This is how basically we work.
Wow, that's so cool. Well, let's see. I got a couple questions here, and then we'll save it for another day. Let's see, Haynes. I just watched a lot of tech. Okay. Hellspan. Hey Pat, you should jump in the Discord again sometime. We miss you there. Yeah, absolutely. I will. We'll definitely try to do that. I'm just gonna wrap this up. Zara, I really
Mhm.
appreciate your time. Thanks for jumping on to the podcast. It was really a
Thank
pleasure
you for
meeting you.
inviting me.
Yeah, and it's just
Same here.
it's so great to hear your inspiration and how you're developing as a scientist,
Thank
you
you
know.
so much. Thank you.
So we look forward to
Thank you.
seeing you, hearing from you in the future and watching
Thank
you
you.
grow into a professional scientist. Well,
Thank you
guys,
so much.
appreciate the time, everyone. Have a good one. Please hit the subscribe button. We were going to be doing some new conversations in the near future, and we got a bunch of new tech stuff. So we're going to do some a little bit more techy, kind of cool transitions, and we'll be learning a lot more about just how to put on a better show and more guests like Zara to come. So everybody, have a good night. Thanks again. Bye bye.
Bye-bye.
Bye bye.
Bye.